Hi, everyone. Today we're going to start talking about Unit 2 in the AP Bio curriculum, cell structure and function. And we're going to first review the different components of the cell. And as we go through these, I want you to pay particular attention to how the structure of the part of the cell is related to its function. Okay, and for every organelle in the cell, you should be able to describe its structure and the function of it and relate that structure to its purpose inside the cell. All right, so let's start small and let's start talking about ribosomes. Now, ribosomes are structures that are made of protein and ribosomal RNA or rRNA. There are no membranes in ribosomes. Ribosomes are the only organelle in the cell that don't have membranes. Their function, of course, is to use the code in mRNA to manufacture proteins. And something really key about ribosomes to remember is that ribosomes are found in all forms of life, including bacteria, including prokaryotes. So this is considered a piece of evidence for the common ancestry of all life forms. I mean, after all life forms, even bacteria, need to make proteins. So all life forms have their own ribosomes. Okay. And next, let's move on to the endoplasmic reticulum, or ER for short. Now, this and the other organelles we're talking about are membranous organelles. They have membranes. The structure is a series of membranes. They compartmentalize the cell. They divide the cell up. They can also provide structural support for the cell. And there's two main kinds of endoplasmic reticulum, rough ER and smooth ER. Now, rough ER we'll probably spend more time on because that's endoplasmic reticulum that has ribosomes. So a lot of protein synthesis inside the cell happens on the rough ER. The smooth ER does not have ribosomes, and it has a couple of purposes. It synthesizes lipids, detoxifies poisons in the cell. Also, these ER membranes kind of form like little channels, or you can think of them as being like highways inside the cell. And through these channels of ER, materials can be moved throughout the cell, and that's called intracellular transport. Intra meaning within. So ER structure provides support, divides the cell up, involved in transporting things across the cell, can be involved in protein synthesis, can be involved in lipid synthesis, can be involved in detoxifying poisons inside the cell. Next, we have the Golgi. Now, the Golgi looks kind of like an oddball shape of flattened pancakes, except not all the pancakes are the same width. So it's a series of flattened membranous sacs, and the function of the Golgi is to fold proteins and modify them and package them for transport. By the way, proteins made in the rough ER will bud off in little vesicles that will pinch themselves off from the rough ER filled with the protein. Then the protein travels to the Golgi, and that vesicle melds with the membrane of the Golgi, and then the Golgi can modify, like glycosylate, which means add a sugar, make different modifications to that protein. And once that protein's ready, it will bud off in a vesicle from the Golgi. And as you can see in this diagram here, those vesicles will travel to the cell membrane and meld with the cell membrane and secrete those proteins out of the cell. So the Golgi is really important in protein packaging and secretion. Another membranous organelle are the lysosomes. Now, the lysosomes are just membranous stacks that have what we call hydrolytic enzymes. And if you remember from the chapter on biochemistry, hydro means water, lysis means breaking. Hydrolytic enzymes use water to break things down. So the function of lysosomes, they can be used for a lot of things. They can use to break down food inside the cell, break down waste products, break down organelles. They can recycle organic materials in the cell. And don't forget this, lysosomes are also involved in a process called apoptosis that we'll talk about more in Unit 4. Apoptosis is what we call programmed cell death. It's like one of the reasons a cell might want to use apoptosis is that if a cell acquires a mutation that could potentially turn it into a cancerous cell, the lysosome could release its enzymes inside the cell and kill that cell so it doesn't have a chance to grow into a tumor. Okay. And then finally, for today, we have the vacuole. And the vacuole is just a membrane bound sac. Okay. And it can store a bunch of stuff. It has a number of different storage functions. It can store food, it can store water. In plants, the amount of water inside the water vacuole kind of determines how turgid or how flaccid or wilty the plant cell is. If the water vacuole doesn't have enough water in it, the plant's going to just start to wilt. When it becomes rehydrated and that vacuole is really full, then the plant will become firmer. The vacuoles can also be used in the storage and release of macromolecules from the cell and the storage and release of cellular waste products. Okay, so that's our introduction to the start of the cells. I hope that makes sense. Have a great day, and I'll see you next time.